Hello everyone, my name is Raiyah Patton, and the topic that I'm going to be discussing with you all is the role of VEGF-C in regulating lymphatic endothelial cell barrier function. Some important information that we should know is that the lymphatic system is actually a vascular network, and what it does is it functions in maintaining the balance between tissue and fluid homeostasis within our cells. These lymphatic vessels are actually composed of the lymphatic endothelial cells, and these cells actually help form and support the semi-permeable barrier, which separates the lymph from the interstitial space within our cells. These lymphatic endothelial cells actually regulate the amount of fluid that's able to be transported between the inside and the outside of our cell. Although this important role is known, there's actually still a lot to be investigated about how the specific molecular mechanisms control the permeability of the lymphatic endothelium. Next, if we look at Figure 1, Schematic A, here we see lymphatic vessels, and as we discussed before, they are composed of lymphatic endothelium. Here, these lymphatic vessels are actually intermingling with the blood vascular network. Next, if we look at Image B, here we see VEGF ligands and their respective receptors. Here we see that VEGF-A actually prefers to bind to VEGFR2, and this occurs primarily in blood endothelial cells. Next, we see that VEGF-C is actually capable of binding to VEGFR2, although it primarily binds with VEGFR3, and this occurs in lymphatic endothelial cells. From previous research, it's found that VEGF signaling is actually imperative for lymphogenesis and angiogenesis to occur. Next, if we look at Figure 2, here we see a schematic, and it's demonstrating the regulation of VE cadherin by the activation of VEGF-A and VEGFR2. Next, if we look at Image B, here we see two mouse embryos. The first mouse embryo is actually VEGF-C, meaning that it lacks the presence of VEGF-C gene. The second mouse is heterozygous, meaning that it does have one allele of the VEGF-C gene. Here we can see in the VEGF-C knockout mice, there's actually significant edema and slight hemorrhaging. However, in the mice that does have VEGF-C, we see that the mouse is a lot more normal in size, and we don't really see any hemorrhaging. Next, if we look at Image C, here we see cells that are VEGF-C knockout, and we see that these cells actually fail to form lymph sacs. However, the cells that do have VEGF-C were able to form lymph sacs. In addition, the VEGF-C knockout cells, we can observe some hemorrhaging, which is displayed in the red of the first image. Furthermore, in blood endothelial cells, endothelial permeability is typically controlled by the phosphorylation of vascular endothelial cadherin, or VE cadherin. And this actually happens in response to the binding of VEGF-A to VEGFR2. While it's evident that VEGF-C is very important for lymphatic development, as we discussed in Image B and Image C, here it's actually still unknown how VEGF-C can affect vascular growth as well as the permeability of cells. Furthermore, the objective of our study was to investigate how the role of VEGF-C works in the phosphorylation and localization of VE cadherin through lymphatic endothelial cells. Furthermore, the method of which we proceeded to obtain our results is displayed in the schematic in Figure 3. Here we can see that the lymphatic endothelial cells were actually treated with VEGF-C, and we actually monitored these cells over time, recording our data at the 1 hour, 3 hour, and 6 hour time points. These cells were actually fixed with 4% PFA and stained with antibodies. We used immunofluorescence imaging to visually analyze our results. We used Fiji image analysis to actually quantify our results. And then we took our numerical data and we graphed it so we can easily see the correlation between our results. Next, if we look at Figure 4, here we see a representative immunofluorescence image of the VE cadherin, which is displayed in red. Here we can see that there were significant changes of the VE cadherin over time. We see that the VE cadherin did fluctuate in thickness, however, it did overall decrease in time. In addition, the VE cadherin did become increasingly less continuous over time. Here, if we look at Image B, we see that the VE cadherin thickness did fluctuate significantly, but overall decreased. Then if we look at image C, we see that the amount of breakages within the VE cadherin significantly increased over time. Next, if we look at figure A, here we see a representative immunofluorescence image of the phosphorylated tyrosine 658 VE cadherin. Here we see that these cells that were treated with VEGF-C, we can see that they actually became increasingly more phosphorylated, and the phosphorylation of the VE cadherin continued to move further into the cytosol. Next, if we look at figure 6A, here we see a representative immunofluorescence image of the phosphorylated tyrosine 685 VE cadherin. Here we can see as time progressed, the VE cadherin continued to be phosphorylated and continued to become relocalized further into the cytosol. However, we did notice at our 6-hour time point, our numerical data was actually not significant, and this was likely due to our small sample size. Overall, the primary conclusions from our experiment that we were able to obtain were that we can in fact demonstrate that the treatment of lymphatic endothelial cells with VEGF-C does result in changes into the VE cadherin morphology, as well as a decrease in their continuity. Interestingly, there actually was initial thinning of the VE cadherin junctions after treatment with VEGF-C at 1 hour before we could actually notice an increase in thickness, and this is displayed in image B of figure 4. Another finding was that the treatment of lymphatic endothelial cells with VEGF-C did result in the phosphorylation of VE cadherin at location 658 and 685, and this contributed to their relocalization further into the cytosol. Another additional finding was that the phosphorylation of VE cadherin at location 685 was actually not significant at the 6-hour time point, and as we discussed, this was likely due to our small sample size. Furthermore, the changes in the VE cadherin likely were related to the changes in the endothelial resistance, and this could potentially be the mechanism by which the permeability of these lymphatic endothelial cells is controlled, because we know as resistance decreases, the cell becomes more permeable, making it easier for the phosphorylated VE cadherin to localize further into the cytosol. Overall, this mechanism could be related similarly to how VEGF-A and VEGFR2 regulate VE cadherin in blood endothelial cells. Some future directions that we would take for this experiment would be, we would like to further investigate the mechanism by which VEGF-C actually results in the phosphorylation of VE cadherin, because this still actually remains unclear. Another direction we could possibly take would be to focus on measuring the actual effects of the VE cadherin phosphorylation on lymphatic endothelial resistance. Another future direction that we would be interested in pursuing would be to see if mouse models could further inform us of the physiological role of VEGF-C. At this time, I would like to acknowledge and thank the members of the Con Lab for their helpful discussions, particularly Patricia Marico, who was our lab manager, and I would like to thank the American Physician Scientists Association Virtual Summer Research Program for allowing me to have this wonderful opportunity. Thank you for watching.